Okay, so cognitive map, what do we mean by that? Just to remind you of this classic study from the 1940s in rats, where the rat, when they learned this route and then went up here and found their goal block, the rat immediately comes out and goes straight toward the goal, telling you they've learned something much more interesting than the series of left and right turns to get to the goal. They must have done something much more like actually learn the layout of space and the relative position of that goal so they could come up with a new vector to get there when the original route was blocked. Okay, and you guys can do this too, right? When your route is blocked, you come up with a novel route, right? And you do that by having some knowledge of your environment, something tantamount to that in your head, some version of that. And further, you know where you are in that map. Like right now, you know where you are, okay? Now here's the cool thing. Specific neurons in your hippocampus right now are firing, telling you that you are right there. Okay? So these neurons are called place cells, and this is what they do. Okay, so I'll be a place cell, or what I'll do is I will act out the activity of a place cell by a series of clicks I will make as I walk around. So imagine there's an electrode in my hippocampus, and you are hearing the activity of a single neuron in my hippocampus as I walk around. And here's what it would do. You'd hear background firing, so it's going to go click, 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 noisy background firing, click, 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 click. Okay, I'm not going to go down there. Click, 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 click. So that's one neuron that fires only when I'm right over there, that place. It's not where I'm facing over there. It's not what I'm looking at particularly. It's when I'm right there. Okay? That's a place cell. And so there's lots of place cells in your hippocampus that do that, and they do it for different locations in your environment. And all of this was first worked out, of course, in rodents who were running around, who had electrodes in their hippocampus, but where those electrodes were connected with a loose tether so that the rodent could move around in their environment while recording from individual neurons in the hippocampus. Okay, so that's the setup. And so I'm going to show you a movie of an aerial view of a rodent moving around, a rat moving around in its environment. Can you see the little rat there? And what's happening is this video is tracing out the rat's path with the light gray. And every time that, it's recording from one neuron. Every time that neuron fires, it makes a red dot. And so this is obviously sped up. But as the rat moves around in his environment, you see an accumulation, like more firing when the rat is right there. It's not which direction the rat is going through when he goes there, just basically whenever he passes through that in any direction, neurons fire more than anywhere else. And then if we take that and blur it, as scientists like to do to make nice idealized pictures, that is the place cell for that neuron. That is the place in space that animal has to be to make that neuron fire. Okay, right. So in answer to Shasha's question, here are a bunch of place cells from a rodent exploring the same environment. So you might say, well, there's like a hotspot here and a little sub one there. But in general, most of these cells respond with a hotspot in a particular single location in this particular environment. Okay? Did you have a different question about that?
Yes. So that all depends on the rat being conscious of the fact that it's in that place?
If the rat was anesthetized, or if he was, blindfolded and you passively moved him around in that space and he had no idea, no way to tell where he was, that wouldn't work. However, if the rat knows the environment and then you do this in a darkened room where he's actively locomoting around, these things will still work pretty well, because rats are very good at keeping track of where they are, even without visual cues, if they know the environment. They'll have other cues, like tactile cues, and they will know how far they went in each direction. Remember I talked briefly about the Tunisian ants doing dead reckoning? Right, keeping track of their vector and speed at each moment and integrating the whole thing to know where they are. That's called dead reckoning. Rats are pretty good at that too. Let me say a little bit more about this. So just to cash this out, the place field is the location in space the animal has to be to make that hippocampal cell fire. Okay. Okay, so what about, we and rodents tend to go around mostly on a 2D plane. That is, we have buildings and trees and stuff. We sometimes go up in the z-axis, but mostly we live in a 2D plane. But that's not true of all animals. So recall the bat that I mentioned last time, these amazing flyers and navigators who fly in 3D in complicated trajectories and yet have amazing abilities to keep track of where they are over 30 to 50 miles that they fly at night, and even as they change their orientation. Well, it turns out that in the hippocampus of bats, there's a bunch of work where people have put remote, what do you call these things? Recording devices on bats where you can remotely record neural activity in the hippocampus as the bat flies around. And turns out that bats have place cells too, and their place cells, they also can do this in a lab environment where they're flying around and you keep track of their location with cameras, so you know exactly where they are in 3D space. And it turns out that place cells in bats are three-dimensional, because bats live in a three-dimensional world. So whereas these would be a bunch of kind of schematized place cells of different hippocampal cells in a rodent, these are different place cells for different hippocampal cells in a bat. Make sense? Bats need this. Okay. So I said before that I had one of those and I acted it out, but what's the evidence for that? That evidence in humans came way after the evidence in rodents, because as you can imagine, it's harder to arrange to record from individual neurons in human hippocampuses. Nonetheless, as I've mentioned a few times, there are occasional opportunities where a neurosurgeon has stuck an electrode in an interesting part of the brain for clinical reasons, and the patient and the neurosurgeon are nice enough to let scientists collect data. So I'm going to show you a really gross, bloody picture. If that's going to bother you, just look away, okay? This is neurosurgery. You take the skull off, you take the dura off. That's the direct surface of the brain. The neurosurgeons stick electrodes right on top of there, and in this case, they put them deep inside the brain. Okay, the gross pictures are gone. We have just a nice, clean X-ray here. So in these cases, these are, this is a patient who's got an electrode sticking straight into the brain from the surface, straight down into the hippocampus. Okay? And so what would you do if you had a patient who was willing to do your short experiment while hanging out in the hospital waiting to have a seizure with electrodes in their hippocampus? Well, you'd have them play a little game in a virtual space in some kind of, you don't even need VR, you can use a pretty cheesy little video game, and I'm sure this one was quite cheesy. This study was done back in 2003. So they had patients navigate through a space. This is an aerial view of the space. The patients didn't see that. They saw this kind of front view, and they kind of navigated around with the joystick in that space. And there were three kind of visually recognizable kind of locations in that space, and they had to do things to go from one location to another. Okay, details don't really matter. So all the while, Ekstrom and colleagues are recording from individual neurons in this patient's hippocampus, okay? So here's an example of a place cell. So this is a diagram of the space I just showed you, right, with those three recognizable locations and other locations that the patient could virtually navigate through with a joystick. The red lines are the patient's trajectory as they moved around in that space. And the colors within each square are the average firing rate when the patient navigated through that location. And so this is the place field of that individual cell in this patient's brain as they went through this space, because the firing rate there was around 5 hertz compared to 3 hertz for some other locations, and mostly lower than that. Okay, does that make sense? So we're just like the rodent experiment, but it's a person with a joystick looking at this space as they go through this virtual environment, and we're mapping out their place fields like that. Okay, all right. So that's a modern version of a classic problem that animals face in their environment. They may know where they are, but that doesn't tell them which way they're facing. So just to be really concrete about this, so here's an aerial view of a person. You're standing here. You have a cognitive map in your mind, and your place cells are telling you your location in that map. Okay, so you know where you are in that map. But and you're looking down a street, so you know that you're oriented with respect to some external axis like this. But you don't know how your mental map should be aligned with that street. Are you facing like this, facing north in Manhattan? Or are you facing south, right? So that's the problem of reorientation, is figuring out your particular orientation, not just your location, but which way you're facing in a known environment, okay? And we've all faced some version of this, presumably, at some point, and it's annoying. It takes a while to figure out, and then, I don't know if anybody's had this experience. I've had it only in Manhattan, because that's where this arises for me, but I'm sure there are other locations, where you come up and you think you're going one way, and then all of a sudden, like it's like your whole mental map goes kaboom. So how many people have had that experience? Like, it's very sudden and punctate. Yeah. Turns out that when that happens, all of your neurons flip together in unison. Like, they're all in cahoots. They have one version of this. When you have that experience, it's because they're all flipping together, and I'll show you some data on that in a second. Okay, so I just said how all this, I got ahead of myself, how it relates to behavior. But just to go through that quickly. So what we've done here is they've trained the mouse on this classic reorientation task. They disorient the mouse before each trial while recording from hippocampal place cells. As before, given cell flips 180 degrees from trial to trial, despite the fact that the stripes should disambiguate it and tell him which way he's oriented. And by the way, the head direction cells and the grid cells also flip in the same way in cahoots with the place cells. But you can tell which corner the animal will go to by looking at what the place cells respond, right? And so when this place cell represents that location, the animal searches first there, and when it flips around, they search in the opposite corner. Okay, so all of that just shows this really strong link between the place cells and behavior. So to recap, we've talked about 4 different kinds of cells involved in representing space and navigating around in it. Place cells that are like the you are here, they respond when you're in a particular location. Direction cells that respond when you're heading in one direction, not in another direction. Border cells that fire when you're near a particular border in the environment. I have border cells going right now throughout this whole lecture. I've got a batch of border cells, right, that are going. Grid cells that do this amazing thing of firing when the animal is in multiple different locations, and those locations that make it fire are arranged in a hexagonal grid. Think of it as a kind of ruler telling the rodent how far he's gone in his space, and those grid cells are like the rulers. Yeah, right. Those are the 4 kinds we've talked about. Okay, so now here's the cool thing. All this stuff, navigation is awesome, right? We need it. It's important. All mobile animals need it for the reasons we've been talking about. But we can use this whole system for so much more than just navigation. Once you have this fancy system in your head to keep track of your location, to keep track of your direction, to keep track of where things are, how you're moving through that space, you can use that whole magnificent system in other ways. And in the last 3 or 4 years, there's just a huge number of studies that are really starting to take this very seriously, particularly the grid cells, and thinking about how the grid cells, I mean, probably the whole system, but people have been focusing on the grid cells and how they're used in multiple different situations. So here's one. Okay, this is a cool study where what these guys did was they stuck a little device hanging around people's necks, the subject's necks, have a little camera aiming forward. It takes pictures at random intervals and records the person's GPS location, okay? So you send them off for a few months with this little device, and you do something to protect people's privacy. I don't know exactly how they maneuver that, but I'm sure they found a way. And so then they get this set of photographs taken from this person's kind of front view of wherever they were over several months as they went wherever they went in their lives, okay? Okay, so now what they do is they use multiple voxel pattern analysis in the hippocampus while people are reliving those experiences in the scanner by looking at those images taken from their front-facing cameras, okay? And then they asked, is the pattern of response in the hippocampus, like some bunch of voxels, here's some pattern, is it more similar for events the subject remembers that were nearby in space, okay? And they were. So this is the distance on a log scale between two patterns that result from the subject looking at two different images. And this is the similarity of the pattern in the hippocampus. Now, get how it's telling you that the hippocampus is remembering and reliving some representation of the locations of those of where you had those experiences. Everybody get how this shows us? But then they asked another interesting question, and they said, oh, does it also represent time? So we've been talking about space for the last 2 lectures, but now we're like going straight off the deep end. And our first step, not even near the deep end yet, is does it do not just space but time? So they can take all those photographs and say, okay, how far apart in time were these 2 photographs taken? And they can do the same graph. And yes, they get a relationship with time as well. The farther apart in time people saw those 2 patterns, the more different the patterns in the hippocampus. Isn't that cool? Okay. So that's one example showing that the hippocampus holds some kind of large-scale representation of not just space but also time. And so there's a lot of work on how this gives structures to our memories for distances over the range of 100 meters and times between 15 hours and a month. Okay, so that's the first thing. Here's another even more radical example. Okay, so people have shown that grid-like representations, and I'm skipping over most of the details here to give you the gist, because actually the details are a bit complicated. But they've shown that people seem to use their grid cell system when they are thinking about conceptual spaces, not just physical spaces. In another recent study, they had subjects do a role-playing game while in the scanner. And in the role-playing game, they're interacting with virtual characters, and those virtual characters had different kinds of social power and different affiliations with other individuals. So here's another kind of the social space that was kind of invented by the experimenters, and the subjects are playing this game, interacting with other virtual individuals who vary in social dominance and their affiliation to others. And they find place cell activity that seems to echo the position of another person in that social space. I mean, that's extremely abstract. And yet again, parts of the navigation spatial system are being co-opted to do this. Here's another very charming non-spatial use, well, sort of spatial use of place cells. So those bats, turns out, are extremely social organisms. They have very sophisticated social structures, and they care a lot about each other and who's related to whom and who's doing what to whom. And it turns out that there are social place cells in bats. That is, cells in this bat's brain, if I were a bat, that would be representing your location, Jack. Okay, so not the usual thing where my place cells are just saying, where am I? I'm watching you, and my place cells are telling me, where are you? Right? Something social organisms care a lot about, including bats. So they have an observer bat here hanging upside down, and he's watching this bat fly over to there and back, and then in this experiment, he subsequently flies that same path. That's how we know that he's watching that path, because he has to mimic the bat's path that he just observed. But while he's watching that bat fly on that path, what you see is here's a cell right here. Here is the path flown by the bat, like out and back. And this is when the bat is flying out and back himself, and this is when the other bat is flying out and back. Blurred a little bit, here's the place field for self and the place field for other. They're not the same. A given cell doesn't represent the same location when it's me who's there and when it's the person or bat I'm watching who's there, right? But they're place fields in both cases. It's used not just for representing social status, what kind of bird this is in this abstract bird space, but actually for making decisions, for thinking. Okay, so as rats run in mazes, you can record, we've shown this already, you can show multiple hippocampal place cells. And can you guys imagine that if we were recording from several different hippocampal cells at the same time, we could read out those cells and make a guess about where the rat is in its location, right? It's just like MVPA, but done across neurons, right? So we have a pretty good sense of where the rat is, okay? So now we have a rat navigating around in this maze, and what I'm going to show you, the white circle is where the rat actually is, and the little color thing is telling you where the simultaneous readout from several place cells in that rat's hippocampus would predict where the rat is. Like, can we tell where the rat is by looking at his place cells? Okay, so right here they're in the same place. It makes sense. The rat is right there, and we're reading it out. Okay, so far so good. But now what we're going to do is watch what that place cell location does as the rat moves around in his environment and makes decisions about where to go next. Okay? Okay, so what we're going to see is the rat is going to come up to an intersection of the maze. I think it's I think it's right here. And he's going to decide, am I going to go this way? Am I going to go that way? And as the rat stays there deciding which way to go, and the white dot stays there as he sits there thinking, should I do this? Should I do that? You could call it neural deliberation. We will see what his place cell activity shows you. Okay, so here we go. Rat starts there. Whoops, how do I play this here? Okay, so rat is heading up there, and so are his place cells. He comes up to the intersection. He stays in one place. But look what his place cells are doing. Should I go over there? I'm just interpreting what this means. But it sure looks like neural deliberation to me. And that's what he decided. Everybody get what we just saw? While he's standing there, he's in one place, but he's clearly deciding where to go next. And while he's deciding, those place cells are essentially, apparently, running simulations of where he might go next.